Good day everyone. Welcome to our presentation for SIR2031 on food microbiology. We are excited to share our finding and insight with all of you today. Our presentation focuses on identifying the foodborne pathogen Vibrio Parahaemolyticus from suspected food items. This bacterium is a significant concern in food safety and public health. Before we dive deeper into the topic, let me introduce our teammates. First is Abida Armin, Mim Shali Krishnamurthy, Noor Shamiza, and Tan Ejen. We look forward to discuss this topic in detail with all of you. Vibrio Parahaemolyticus is a J-shaped halopic gram-negative float that belongs to the family of Vibrionaceae. It is commonly found in uterine and marine environments, and it's particularly abundant in warm coastal waters during the summer months, making it a significant concern for seafood safety. This bacterium is a known cause of foodborne illness, primarily resulting from the indigestion of contaminated seafood, especially raw shellfish. Infection can lead to symptoms such as fever, nausea, abdominal cramping, and diarrhea, while gastrointestinal illness is most common. Vibrio Parahaemolyticus can essentially cause more severe conditions like septicemia or wound infection. Understanding these properties and risks is essential for the prevention and management of infection caused by Vibrio Parahaemolyticus.
We are representing Vibrio Parahaemolyticus, morphology and structure. It is a gram-negative bacterium which is of 0.5 to 1 micrometer in diameter. The shape of it is straight or curved rod with a single polar flagellum. Environment adaptability. It is halophilic, growing in 0.5 to 10% NaCl and thrives in aerobic condition. It is mostly found in salt water and grows best at the temperature between 10 to 45 degrees centigrade, with an ideal preference for 37 degrees centigrade. It can survive in a pH of 4.8 to 11. Now we will be discussing about Vibrio Parahaemolyticus biochemical characteristics. It is oxidase positive, sucrose negative, and facultative anaerobic. The outer membrane contains lipopolysaccharide which is LPS contributing to virulence, produces pili which facilitates attachment to host cell and surfaces. Survival mechanism. It has several ways to survive harsh condition. It forms biofilms to protect itself, produces a harmful toxin called thermostable direct hemolysin (TDH), and can survive both cold temperature and high salt level. Additionally, it uses genetic mechanism like quorum sensing and biofilm formation to adapt and overcome challenges that threatens its survival.
Now, I will be discussing the significance of Vibrio Parahaemolyticus in suspected food sources, specifically in boiled croakers. Boiled croakers are considered a high-risk food because Vibrio Parahaemolyticus is naturally present in the marine environment. The risk increases due to the factors such as poor cleanliness, improper storage, and undercooking, which can lead to food contamination. Vibrio Parahaemolyticus contamination can cause gastritis, characterized by symptoms such as diarrhea, abdominal pain, and vomiting. To prevent this, we must ensure that seafood is properly cooked, avoid consuming raw or undercooked croakers, and maintain hygiene, storage, and handling practice. These steps are crucial in reducing the risk of foodborne illness associated with this pathogen. Now, I will be presenting on the outbreak caused by Vibrio Parahaemolyticus. Let's start with the primary source of this pathogen, raw or undercooked seafood. Vibrio Parahaemolyticus is a marine bacterium commonly found in the warm coastal water. It is often associated with shellfish like croakers, clams, and oysters. In this case, the risk is partially high for kerang rebus or boiled croakers, especially if they are undercooked or not handled properly. Another key source is contaminated water. The bacterium thrives in breakage and seawater, especially during warmer season. Seafood or other food washed with contaminated water can easily become a transmission vehicle. Poor hygiene and cross-contamination also play a significant role. Improper handling of raw seafood or the use of shared utensils can lead to contamination. Food handler with inaccurate hygiene practice can exacerbate the problem. Now, let's have a look of temperature abuse, which is another critical factor. Vibrio Parahaemolyticus multiplies rapidly when seafood is not refrigerated or stored at appropriate temperature. Improper storage of boiled croakers could have allowed the bacteria to thrive. Environmental factors such as warm climate and high humidity also contribute to increased contamination risk, particularly in outdoor festival settings. Another important aspect to consider is the pertinence of questionable food products. The principal source of contamination in this case is seafood, particularly cooked croakers or kerang rebus. Seafood contamination poses a significant risk due to improper handling and storage. Additionally, there are other hazards to consider. For instance, the processing of food or dessert like chendol may be impacted if contaminated water is used during preparation. These factors highlight the importance of strict hygiene and proper food handling practice to prevent foodborne illness. So next, we will be explain about to design a testing protocol in order to detect and isolate the pathogen, which is Vibrio Parahaemolyticus, from the suspected food items such as boy cockles. So we will be suggest laboratory methods to confirm the presence of the pathogen, and also we will compare the standard protocols such as ISO and BAM for detecting the pathogen. So this is the example of the image violet colonies of Vibrio Parahaemolyticus on chrome agar Vibrio agar plate. Okay, so I will be present about the methodology to isolate and detect the Vibrio Parahaemolyticus from the suspected food item, which are boy cockles. So this methodology is divided into 6 parts to isolate and detect the Vibrio Parahaemolyticus in order to confirm the presence of the pathogen. So firstly is we will collect the samples of the cockles, and then we will take or weigh 25 grams of each cockle samples to homogenize in 225 ml of alkaline peptone water. So the purpose of the homogenization in alkaline peptone water, it can ensure that the bacteria are evenly distributed throughout the sample, which can increasing the likelihood of detecting Vibrio Parahaemolyticus. Next, we will prepare a 5 dilutions of the MPN series by using 3 tubes per dilution as described in the FDA's bacteriological analytical manuals to determine the total of Vibrio Parahaemolyticus density in the samples. Then, we will conduct the enrichment culture by transferring 1 ml of each broth culture into a sterile tube containing 9 ml of salt polymyxin broth and incubate overnight at 35 to 37 Celsius. The purpose of using enrichment broth of salt polymyxin broth is it can selectively enhance the growth of Vibrio Parahaemolyticus while suppressing unwanted microbes. It also can further enriches the culture by providing conditions that favor for the growth of Vibrio Parahaemolyticus, which can make it more effective to isolate the Vibrio Parahaemolyticus. After that, we will conduct isolation on selective agar by place a loop full of culture from each probe culture onto chrome agar Vibrio and incubate overnight at 35 to 37 Celsius. Then, we will observe and identify the typical violet colonies formed on the agar plates, which indicate the Vibrio Parahaemolyticus. So, the use of the chrome agar Vibrio, or known as CV agar, can allow the selective isolations of Vibrio Parahaemolyticus that are easily distinguished and differentiate from other Vibrio species, such as the form's colonies is violet color, which make it easier to identify the Vibrio Parahaemolyticus. Okay, next, we will conduct the DNA extraction. So firstly, we will inoculate 1 violet colony from the agar plate into 5 ml of Luria bertani, or known as LB broth, and incubate overnight at 37 Celsius with shaking at 120 rpm. Then, we centrifuge 1 ml of the overnight culture in LB broth or cell culture from 6-hour enrichment broth at 10,000 G for 5 minutes. After centrifuge, discard the supernatant and resuspend the pellet in 1 ml sterile distilled water. Then, boil the resuspended pellet for 10 minutes to facilitate the cell lysis and DNA release. So, by boiling the cell pellet, it releases the cells and release the DNA into the supernatant, which is then to be used as a template for the PCR, which is necessary for the subsequent PCR analysis in order to confirm the presence of the specific genes associated with Vibrio Parahaemolyticus. After that, we will immediately place on the ice for the 10 minutes and centrifuge the cell lysate. And lastly, we transfer the clear supernatant to a new tube to be used as a template in the PCR assay. Okay, lastly, we will conduct PCR assays by preparing the PCR reaction that consists of 20 microliter reaction mixtures. After that, the PCR reaction will be conducting in a thermocycler with the following reaction conditions below that are divided into 2 parts for the TDH and TRH primer and ToxR primers. The primer used for ToxR is to target the genes of VP ToxR, which is for the identifications of the specific species of the Vibrio Parahaemolyticus, while for the primer TDH, or known as a thermostable direct hemolysins, and TRH, and known as TDH related hemolysin, is to target genes that have a TDH and TRH genes that is specific for the presence of the virulence factors of the pathogenesis of the Vibrio Parahaemolyticus. So the purpose of the conducting PCR assay is to identify and detect the presence of the virulence factors that carrying the TDH and TRH genes from the Vibrio Parahaemolyticus. And lastly, we run the PCR products by electrophoresis in a 1.8% agarose gels and stain with ethidium bromide. Then, we will record the results by using a gel documentation system. So, the purpose of the gel electrophoresis is to confirm the presence of the amplified target DNA, such as ToxR, TDH, and TRH genes that based on their size of the PCR products, which also essential for validating PCR results and ensuring accurate identification.
Hi, my name is Ijin and I'm going to talk about detection of Vibrio Parahaemolyticus in cockles by ISO and BAM. Internal Organizations of Standardization, which is ISO, are the standard method for bacteria detection in food. Bacteriological Analytical Manual (BAM) are laboratory procedures for microbiological analysis of food and cosmetics. ISO and BAM are standard methods that can be used to detect the presence of Vibrio Parahaemolyticus in cockles. However, when compare ISO and BAM with the rapid detection method such as PCR, ISO and BAM are more time-consuming, laborious, and high caustic. Next, I'm going to talk about the differences between ISO and BAM. These two methods involve morphological tests and biochemical tests, such as oxidase tests and sugar fermentation tests. Both of them use similar culture media, such as thiosulfate citrate bile salt sucrose as plating media. The differences between ISO and BAM are ISO can be used internationally as it is standardized, whereas BAM are used more in laboratories, especially in United States. ISO methods are less flexible and follow strictly to international guidelines, whereas BAM methods are more flexible based on the lab protocols and different agencies. For ISO method, enrichment of culture is done in alkaline peptone water at 41.5 degrees Celsius for 6 to 7 hours, whereas for BAM method, enrichment of culture is done in alkaline peptone water at 35 to 37 degrees Celsius for 18 to 24 hours. Example for ISO method is ISO 21872-1 2007, and the example for BAM method is FDA BAM Chapter 9 Vibrio.
In conclusion, one of the major foodborne pathogens associated with seafood consumption is Vibrio parahaemolyticus. This bacterium poses a significant public health concern due to its ability to produce protein, toxic, and its remarkable adaptability to survive in warm marine environment. Understanding the property and pathogenicity of Vibrio parahaemolyticus as well as the method to prevent its spread is essential in combating infection caused by this pathogen. By implementing associated food safety measures such as thorough cooking of seafood, proper handling, and maintaining good hygiene practice, the risk of illness from Vibrio parahaemolyticus can be effectively minimized. These preventive actions are not just important but critical to ensure the safety of seafood and protecting public health. Thank you.
Okay, here is our references. That's all from us. Thank you.